the middle of the day, we'll start doing our exercises, and then as we get into tomorrow, doing more and more of them. So as we go on, we're going to give you some talks this morning on some new programs that we've actually got going within the TPP. Some are not fully incorporated in the TPP yet, but we're going to give you how we use those. You can also download those and incorporate into your bioinformatic efforts. And then we'll also talk about some resources as we go through. But this morning we have two components. One is the St. Peter, which we spoke about yesterday, and also Kojak. So this is our revised schedule at the moment. So we are going to start at 8 o'clock every morning now. So we have our first one today with Mike Hoopman. And then we also have the data analysis tutorials both on the Sunday and the Monday, both starting at 8 o'clock, where we'll go through what we've learned so far in the TPP, and then also what we're learning today in terms of cross-linking and also some other tutorials as we go through. So these are going to be fairly hands-on type days as we go through. So today we're going to finish off with what we have in terms of programs, go into some of the resources that we have, and also talk about some future aspects as well. So our speaker this morning is Dr. Michael Hoopman. So Mike, who you've seen here and also had him talk so far, is both a software engineer as well as a scientist. He has a quite extensive background in protein analysis, especially with high-resolution mass spectrometry, and has been guiding the tools development given his experience in the analysis of these types of proteomes as well. And so at the moment, what we're going to talk about, so we've gone through the TPP process now where we know how to convert the data, we know how to use the algorithms that we're doing to analyze this data, what the benefits are of doing statistical analysis of what we have, and then we also have these programs that can go do this iteratively to get more out of your data. We've learned about the standard file formats that have underpinned all of the Atlas pipeline at the moment, but now we've got a couple of components that we're building, and we're also incorporating them as we go through. And so St. Peter will be the first one we talk about, and then Kojak is really what we're talking about this morning. And then we'll lead off into these other ones as the talks go through. And Mike will take you all through those. Now, the other component that Mike has been doing is working on how does someone actually take a protein complex now and work out that spatial interaction between each other. And we do this by chemical cross-linking. This is just an artistic depiction here. So this is really a crystal structure or a protein structure of three separate proteins bound together. And these are these little tether points where cross-links can be put onto those proteins where you have a free amine, such as a lysine. But if you want to try and work out what that spatial interaction is, does that protein actually sit here, or does it sit here, or does it sit here? We use this protein cross-link and say if there's two peptides, if they're bound together by a cross-link, they must be close together. So that gives us this capability of working out what the 3D structure is of proteins. But to do that at the mass spectrometry level is very, very difficult. You have lots of peptides being produced, and you also have cross-links on those peptides, or even just a single chemical entity. And you have to work out from the mass spectrometry information which one of these is actually being produced. And it becomes a combinatorial problem in terms of bioinformatics. And the issue here is that if you try and do this with very large sets of proteins, you have such a computational problem that it almost becomes impossible. And that's what's shown here, that if you're doing this for a large set of peptides, the more peptides you have that are cross-linked, the computational time just really starts to skyrocket. It's an exponential problem. And what Mike has been able to do is develop a two-pass process with a program he now calls Kojak. And what that does is it searches all of the data sets and tries to derive initially from these peptides which are the best ones to look at, and then also works out what that fragmentation pattern is, and then gives you identification. So what happens here is it's more of a linear computational expanse rather than exponential expanse. So if you're trying to do this with larger data sets, that you get a lot of benefit here, that your analysis is done very fast as compared to doing this very slow, and so you can get a lot of this analysis being done very quickly. And so with that, I'll leave Mike to tell you all about the intricacies and how to get this done. So if people are interested in cross-linking, this is really a program that you want to be able to use. Mike?
All right, good morning, everyone. As Rob introduced, I will be starting this morning's lecture. And so we are actually going to take a step back and talk a little bit about database searching again, but using a specialized algorithm specifically for detecting protein crosslinks. And so this is a big departure from my last lecture. But it highlights just some of the latest techniques and some of the most interesting techniques being applied to shotgun mass spectrometry today. And that's going to be analyzing cross-linked proteins. And again, the outline for today is I'm going to go over briefly protein interactions and how they are measured in a mass spectrometer. I'm going to tell you how cross-linked proteomics data is interpreted, give you an overview of the Kojak algorithm, and then the tutorials and exercises are included in this, and that we'll get to use Kojak, just not today. It'll be 2 days from now. We will go over the actual tutorial, but I wanted to get everybody on board with how the algorithm works, and so that when we do the exercises, you'll be able to jump in quickly and start analyzing data. So protein structure is, of course, a very important field of biological analysis, in that, in particular, the function of any given protein is often dictated by its structure. And so therefore, a lot of researchers are interested not just in knowing that a protein is in a sample, but actually what it looks like and how it interacts with other proteins. And then even more importantly is that they would then consider how that structure changes with relation to disease, or how they can manipulate that structure to, say, develop therapies, custom enzymes, and do other, and alter biological function within a cell. So shown here, we take our protein structure from the primary structure, the series of amino acids, into the beta sheets and alpha helices of the secondary structure, which get assembled into this tertiary structure, or what individual proteins look like when properly folded. And then finally, those combine into quaternary structure, or complexes of proteins that interact with each other to create new or supplement function that is not possible with the tertiary structure alone. And when we do shotgun proteomics, usually what we do is reduce proteins from this level here all the way down to the primary structure, and use just the primary structure for protein identification, and in some cases even quantification, as you learned today. But we never trace that information back to the actual quaternary structure. And this has been an interest of a lot of mass spectrometrists for over a decade now, I think, and they've always kind of considered, are there ways that we can get at protein structure from a mass spectrometer? Because a lot of the other methods that are available to them, such as X-ray crystallography or NMR spectroscopy, are expensive, difficult, and very time-consuming. And especially for X-ray crystallography, not all proteins can be crystallized. And without crystals, they actually can't obtain structural information. So computational prediction algorithms exist, in which various researchers try to just take an amino acid sequence and predict what the structures would look like. But these have their own problems, in that they're very computationally intensive, and often they require some homologous tertiary structure, to start their algorithm in the right path to produce an unknown tertiary structure. And therefore, they are also limited in the number of proteins that can be studied this way. So mass spectrometry hopes to circumvent these issues, and I'll go into detail as how that is done. As Rob has shown you this slide already here, as you can see, you can take any protein complex of interest and chemically cross-link it. And I'll go over that in a moment, but these cross-links create tethers that then bind when they pass between two of the protein subunits in the complex, such as this blue subunit here on the outside and this red subunit tucked in the middle here. These tethers create a covalent bond that after digestion with your trypsin, those peptides remain linked together. So the association between any two parts of the protein, I'm going to switch here and show you this green protein here, tethered on the end to this blue protein here, tells us that in three-dimensional structure, these proteins are proximal to each other, and that after digestion, they remain tethered, telling us that if we can identify these peptide sequences, that gives us information that these are the two specific regions of the intact protein complex that were originally proximal. The relationship between crosslinker and its efficacy, as well as its binding properties of where it can attach to the protein. There are actually many different crosslinkers available that can change the properties of the analysis. And all of that needs to be considered downstream when it comes time to analyze the data. So to give you a close-up example of cross-linking here, I have a cross-linker known as BS3. It's a commercially available cross-linker. It's fairly inexpensive to purchase. It's very commonly used. It has this carbon spacer arm here, which dictates the length of the cross-linker or the maximum distance between amino acid residues over which it will attach to a protein complex. And it has these reactive groups on either end. And this is a homobifunctional crosslinker, meaning these reactive groups are the same on either end. And these groups determine where on the protein the crosslinker will bind. And so with BS3 in this reactive group, it's an amine reactive crosslinker, which means it will only react with primary amines. And primary amines are lysines inside your protein, as well as the N-terminus of the protein. So it'll very specifically bind to lysine. And when it binds to a lysine on one end of the crosslinker, the other end will float around and look for an attachment point to another lysine. And should this point exist within this spacer arm here, which is 11.4 angstroms, a covalent bond is formed that remains in place after digestion with trypsin to produce the peptides. Now, because the lysine here is cross-linked, trypsin will not digest at that lysine. It'll skip it, but it'll still digest at the arginines here or any unlabeled lysines. And so these are the two peptides that are now one molecule that needs to be analyzed in the mass spectrometer. So there are different types of crosslinks that can be formed. What I'm showing here are two pieces of proteins. Imagine much bigger proteins than what I can show in this image. This is just zoomed in really close at the interface between two proteins, this blue protein at the top and this gray protein below. If we cross-link the sample, the cross-linker can only attach to lysines that are proximal to each other in the actual conformed structure of the complex. So in cases where a cross-linker attaches to a lysine and there is nothing for it to attach to on the other end, this will just hydrolyze and become inert, and no bond is formed. And then there are other cases, such as this lysine here, that perhaps because it is tucked into a pocket in the entire protein complex, that the cross-linker is not capable of reaching it, and so it'll remain uncross-linked. after digestion, there are several types of cross-linked products that are produced. So among them, they each have their own names. What we have is called a loop link or self-link. I usually use the term loop link, so you'll probably hear me say that throughout the remainder of the lecture. This is when the cross-linker occurs at 2 points within the same peptide. In other words, there was no additional lysine or arginine to separate these sequences. They remain a single peptide with a cross-link between it. They also have these monolink or dead-end peptides where the cross-linker has hydrolyzed and therefore is inert. And this is like a typical peptide with now a differential modification on it. There is no pairing for this cross-link here. Then finally, we have the cross-links themselves. And these are what researchers are most interested in. And the first one I'll show here is this intra-protein cross-link, in which two independent peptides from the same protein are cross-linked. These are good for determining tertiary structure in a protein complex, but not quaternary structure, which often researchers are most interested in the actual interface between two proteins in a complex. And so you have this inter-protein cross-link here, where one of the peptides came from protein A, and the other peptide came from protein B. So with this in mind, these cross-links are actually, though you're able to easily produce them through bench methods, the difficult part is actually determining the sequence of each of these peptides using search algorithms. And one of the problems that search algorithms have is the complexity of your typical cross-linked workflow. So as mentioned earlier, you can actually choose from many different cross-linkers that all have different spacer arm lengths, different chemistries on the end of the cross-linker, meaning it'll bind to different amino acids in the protein. And so it'll actually create dozens of different ways that you can cross-link a protein complex. You can also incorporate isotopic labeling into these cross-linkers if you have the money for it, which some people use the isotopic labeling to help with the identification of their cross-links from non-cross-linked peptides in their sample. The cross-linking itself can be done on a purified protein complex or on whole cell lysates, and this is particularly difficult. There are very few studies out there that are successful at identifying protein complexes when analyzing entire cells cross-linked together, but they do this on a discovery-based approach to try and identify interactions en masse. Different digestion enzymes, different instruments to acquire the data with different resolutions, and then finally different data set sizes all create this huge amount of complexity to the workflows for cross-linking. And as a result, there have been few good solutions for analyzing the data. And so the algorithm I will present here, Kojak, is designed specifically to accommodate all of these different workflow choices that are made in producing cross-linked peptide spectra. The algorithm itself, though, has a simple singular function, which is the identification of two peptides linked together in the same spectrum. And so Kojak is itself a database search algorithm, such as Comet, Sequest, Mascot, all the other algorithms you learned about on the first day. But it has one specific functionality that those other algorithms do not have, and that's the ability to identify two peptides at the same time in a spectrum. Even algorithms such as Respect cannot do this, that it's an iterative process. And so here's an example of one of those spectra. It looks like any other tandem mass spectra, except for the fact that it's probably a bit noisier. There is a lot more peak information, and that's because both peptides are represented at the same peak intensity in the spectrum. The B and Y ions that are computed from this have a unique characteristic in that if we step along this red peptide here at the top, the one that starts IDA, and we compute the B and Y ion masses, that's down here, they go along computing just as I had shown on day 1 with the addition of each amino acid mass until you get to this B8 ion, which is this lysine here, where now every B ion is the mass of all the amino acids plus the entire mass of the other peptide. And the same is true along the Y ion series. As I step along here, the Y ions are computed as normal, and then past this point, the Y ions are now the mass of all the amino acids plus the entire other peptide that's linked to it. And the same process must be done for the other peptide as well, shown with blue masses here, that the Y ions all have their typical masses as computed as the sum of each amino acid until you get to the linked lysine, and now the mass includes the entire other peptide attached to it. So all of the existing algorithms for database searching do not incorporate this entire second peptide mass in the linkage and the computation of their B and Y ions, and therefore are not capable of solving cross-linked spectra. Then finally, once cross-linked spectra are identified, the goal is to then map out the interactions between all of the proteins in a protein complex, and then those interactions can be used to create a map of cross-links that then can be fed to other structural algorithms that can assemble low-resolution protein structures and infer what the protein complex looks like in a way that might not be possible using NMR or crystallography. So the Kojak algorithm itself, its single purpose is to identify cross-linked peptides for mass spectra. It is flexible, so any cross-linker can be used, just about any digestion enzyme, and it accepts data from different instruments and different spectral resolutions. I'm going to say right now that whatever instrument you use, if you were to do this sort of analysis, pick a high-resolution instrument. It is possible to do all of this at low resolution. Kojak will work, but you can take your exact same sample, put it into a high-resolution instrument, such as a time-of-flight instrument or an Orbitrap, and the same sample will produce twice as many, up to 10 times as many cross-linked spectra, simply because the complexity of the two peptides in the same spectrum are better resolved with the higher resolution. It makes the analysis much easier. Kojak is scalable, so that it'll work with both small and large protein databases. I will go into the mathematical complexity in a little more detail momentarily, but one of the problems with a lot of the algorithms that existed prior to Kojak was that they could only analyze protein complexes of 2 or 3 proteins. They could not handle a complex that had 10 or 20 proteins. They certainly could not handle an analysis of an entire cell that was cross-linked and then analyzed in a shotgun fashion. So Kojak is scalable that it can handle all of this. In particular, we also have support for multi-threading and cluster computing so that you can even apply large data sets with large protein databases. And then finally, the efficiency is what enables all of this analysis possible in the whole process. The Kojak workflow is actually not too difficult. Mass spectra go into the algorithm at the top, where the spectra are then refined in several ways. So this pre-processing occurs, and this is a way to clean up the data as best as possible and transform it into a way that allows for fast analysis. Once those spectra are cleaned and transformed, a protein sequence database is added to the analysis, and this is where peptide sequences will be compared to the actual spectra, and peptide spectrum matches made. Amongst the top scoring, so additional analysis is used to rank and return to you the top scoring peptide spectrum matches that produce your results that can then be validated using other tools, and that information can then be applied to structural algorithms to reassemble your protein structures. So there are two stages of spectral processing that I'll go over. One is precursor ion refinement. The other is fragment spectra simplification. Precursor ion refinement is necessary to accurately determine the precursor mass of your cross-linked peptides. A common problem with cross-linked peptides in a sample, even if you have a purified sample, the abundance of the cross-linked peptides is usually much lower than the abundance of all the non-linked peptides in the sample. And that's because cross-linking isn't a 100% process or 100% reaction when cross-linker is applied to a sample. So your cross-linking efficiency might be very low for a given site due to its availability or any biases of the cross-linker to an easier to identify and react with site. And so for a lot of good spectra that we have, the precursor might actually look like this. What I'm showing here is a chromatogram, so just the elution profile of a given peptide in the mass spectrometer over a short period of time. And for this particular peptide, there might have been a tandem scan event, a selection event for the peptide at this point in time from a precursor peptide that is only resolved like so. Because its intensity is so low, the mass spectrometer hasn't properly resolved all of the peaks in that peptide isotope distribution. So when it comes time for the algorithm to search for an appropriate pair of peptides, it might assume that this first peak here is the monoisotopic mass, and to search for all peptides that fit this monoisotopic mass right here. However, in reality, the precursor should look like this structure here. So instead of having this 865.4 precursor mass, the actual precursor mass is 865.15. So it's a different mass, which changes the area of the database, your protein sequence database, that needs to be searched in order to produce the correct match. And in order to get to this actual value here, Kojak uses a boxcar averaging algorithm to produce a better resolved precursor ion. And what this is instead of looking for the precursor in this single slice of time in this chromatography, it instead takes a window where it selects that single spectrum plus the neighboring precursor spectra around that event, averages them together. In the neighboring spectra, some of these other peaks might have been resolved. If there's any noise in the area, usually the noise is reduced. It's averaged across all of those scans, and so a cleaner precursor signal can be obtained where now the monoisotopic peak, which was missing from the original analysis, appears down here. The correct precursor assignment is made, and the Kojak analysis will produce a correct peptide spectrum pair for this precursor. The second spectral refinement step is the fragment spectra simplification. So this is now at the MS2 level. In that, as I mentioned earlier, your fragment ion spectra are typical. They're like any other shotgun spectra, except a bit more complex because they come from two different proteins. So there's a lot more peaks. In addition, the precursor ions, because they're two or more peptides, each holding charge state, the charge states tend to be higher for those precursor ions. +4, +5, +6 are very common precursor charges for cross-linked peptides. And as a result, when they fragment, they will produce fragment ions with charge states 2, 3, and 4. So fragment ions are usually represented as very complex isotope distributions in your fragment ion spectrum. I've taken this zoomed out full spectrum view here, zoomed in on a very small region, and you can see for each of the fragment ions there is a fairly significant isotope distribution represented. And each of these additional peaks complicates the analysis. So what Kojak does is take these isotope distributions and collapses them to a single peak representation of the monoisotopic mass of those fragment ions. During this peak simplification process, each of these peaks in the isotope distribution are summed together, and so the intensity goes, as you can see on the left here, from 10,000 to 40,000 for the same fragment ions, so that the actual counts of each fragment are still accurately represented after collapsing the information. Optionally, Kojak can go along here and say, well, now that I have simplified my spectra and I've increased the signal intensity for each of these fragment ions, any ion that remains at low intensity that was unaltered might possibly be a noise ion. So you can define a number here, say the top 500 or top 1,000 peaks in any fragment spectra, and keep those and eliminate all the low abundance noise peaks. This spectra simplification, though, can only be done at high resolution. Low resolution spectra do not actually resolve these isotope distributions, and so it's not possible to do this sort of spectral refinement. And this is an example of why it is best to use high resolution mass spectrometry when analyzing crosslinks. It helps simplify that whole process. And then also, if in the case of spectra that have low spectral complexity, using a flat cutoff to say keep only the top 500 peaks might actually eliminate real peaks of important intensity. And therefore, because for these reasons, all of this spectral refinement is parameterizable, so customizable within Kojak. You can actually change the number of peaks you want to keep in your analysis, and 0 to use all peaks, or you can turn off spectral processing if you have low resolution data. So Kojak is smart enough that you can change your analysis to suit your data needs. The actual scoring algorithm inside Kojak is the same scoring algorithm used in Sequest and Comet. As you know, I'm also a developer in Comet, and so I'm somewhat biased to this scoring algorithm. It happens to be very fast and efficient, and it's survived 20 years of research and development, and still is one of the best algorithms out there. The whole process, as I mentioned earlier, transforms your spectrum from the original spectrum here. This is what the spectrum would look like after doing the isotope collapsing and noise reduction. Your actual spectrum here needs to be transformed to a data structure that the Comet algorithm can then quickly analyze to produce a score. What this process involves is taking small localized regions of your original spectra and normalizing the peak intensities so that these over here actual like B2, B3 ions that are real, just not very abundant, you still want them to match well if you can indeed match a theoretical peak to them. And so this small window here is normalized to produce a larger representation of each peak. So they are analyzed at a sort of equivalent level as the most intense peaks in the spectra. Likewise, there is also these areas down here which are negative score contributions in the algorithm. And these occur when you match to areas of your spectra that contain no signal whatsoever. That actually penalizes you. If you expect to see an ion and it's not there, then there is some sort of penalty formed. So the actual transformed spectrum normalizes peak intensities and creates areas that will penalize the score if matches are made to that area. Rob also showed this figure early on, in that the actual analysis of a protein sequence database when doing cross-linked spectral searches is computationally prohibitive. I gave you some of the complexity before in search space with regular search engines, and that choosing an unconstrained search or choosing a lot of differential modifications creates this enormous search space that can slow down your analysis and to a point where it becomes significant in that you cannot complete an analysis. This is particularly true for cross-linked peptides because not only does all of that information apply, all of those enzyme restrictions and differential modifications apply, but also you are searching combinations of two peptides at the same time, which is a quadratic complexity problem. And so as the number of peptides increases in your database, your computation time grows at this exponential level, meaning that for in this case, if you were searching a single spectrum with, just 1,000 peptides, it might not take much time. But if you were doing it for a larger protein database that has 200 peptides, you now have 500 minutes of analysis for that single spectrum. Or if you were using a really large database, such as like the human proteome, it could take 3,500 minutes for that single spectrum, because of all the different combinations of two peptides that are possible to make in the entire database. So as the number of peptides increase, so does your time. And so instead of searching combinations of two peptides, Kojak uses an approach that allows to search single peptides with a dynamic modification that represents all the possible combinations with other peptides. That reduces the complexity back to a linear problem. You are now searching individual peptides when performing the search. And then identifying what that second peptide is a separate process that is very quick. So the actual time is reduced back to this linear growth function typical with your standard search engines, and therefore it is possible to get through large data sets using the Kojak algorithm. So to give you an example of how this dynamic modification mass works, suppose you have a set of 3 different spectra here, and you have a protein sequence shown up here, and you are looking at a particular peptide, such as this one shown here. It starts IIE. It is a tryptic peptide, but let's say you used BS3 as your cross-linker, which is amine reactive, which means no matter what, to do this analysis, you need a lysine to reside inside this peptide somewhere where the cross-linker would attach to. You know the mass of this individual peptide, and you know the precursor masses of each of these spectra. And so the difference between this precursor mass and your known mass is the dynamic mass, or the mass of the unknown second peptide that pairs to it. You can apply this search to each of these spectra. So you're searching one peptide for each of these spectra, and inside each spectra there is a different dynamic mass. And so because each of these spectra is a different molecule that's being analyzed in the mass spectrometer, there is going to be a different partner for this peptide if this is indeed the correct peptide. This process iterates for all peptides in your database. So to give an example, I'm going to zoom in on just one spectrum here, and I'm going to search a bunch of peptides from that single spectrum. So for example here, I have a spectrum with this precursor mass of 2250, and I'm analyzing my first peptide from the database, has a peptide mass of 1134. Therefore, a dynamic mass of 1115 is applied to all of the fragment ions that are computed for this peptide, and those fragment ions are then matched to the actual data set. Those matches produce a score, and this information is stored in a list so that when we move to the next peptide, different sequence, different peptide mass, different dynamic mass that needs to be placed as a modification on this lysine here, which we assume to be cross-linked, produces a different B and Y ion profile and different matches to the spectrum with a new score here. The score is lower than the original peptide we looked at, so we give this one a rank of 2 and we add it to our list. We repeat the process for the third peptide. This is a peptide here that very poorly matches the spectrum. It has 2 possible sites for the attachment. So both of these sites would actually be checked. But the best site, the one that produced the best score for this peptide, was right here at this lysine. Again, a lower score than the others. It has a rank of 3. The entire process is repeated over the entire database. And then for each spectrum, we ask the following questions. Do any two masses in that peptide list sum together to equal the precursor mass? And does the combination of those, if the answer to this question is yes, does the combination of those fragment ions from each of those peptides explain the spectrum and have a good score? And it turns out that if we go through our list of peptides here, we can actually take the top 2 ranking peptides, we can add their masses together, plus the mass of the crosslinker that's holding them together, and produce exactly the precursor mass of the spectrum. We can then take those fragment ions from each of those peptides and align them to the spectrum, and we see that they complement each other very well in that they explain different peaks in the spectrum. They broadly cover all the peaks observed in the spectrum with small exceptions here, which may indeed be A ions or other things not considered in the process. So that is done. And then together, I can take the scores of each of these peptides and sum them together to produce what would be the best paired peptide score amongst all possibilities in this list to explain this spectrum. And what's interesting in this whole process is even though every spectrum is going to have its own list of peptides, and it's up to the user to define the number of peptides that they keep here. Usually we keep 500 or 1,000 of these peptides for each spectrum. It turns out that in the end, the 2 peptides that sum together to equal the precursor mass are often amongst the top 10 in rank, usually the first 2 in rank, meaning that each individual peptide is would easily provide adequate explanation for approximately half of the spectrum that is observed. And so the whole process actually becomes very quick and accurate. That in the end, what Kojak produces is this list of PSMs here. It's just shown in a text file in any sort of spreadsheet here. We have scan number, retention time, and then we have all of the PSMs that are listed, the best scoring PSM for each of the results. Now, it's always the best scoring PSM, but not always the correct one, because as you know, everything needs to be validated, and we use typical decoy FDR validation, the same as you have learned with all of the peptide and protein profits algorithms so far. And so the way that this is done is simply by providing decoy proteins in your cross-linked search database for the algorithm. So those decoys, any sort of cross-link that is to a decoy peptide is a false peptide spectrum match. We can model the distribution of the decoy matches to the remainder of the PSMs, and then apply statistics and find an appropriate FDR cutoff. The process is complicated a little bit in that for each type of crosslink, remember I explained these at the beginning of the lecture, they have the non-linked peptides which contain, if they contain crosslinker, it's one of these mono-linked crosslinkers that doesn't have a peptide pair to them. Or those loop links, the intraproteins and interprotein crosslinks. Each of these types of crosslinks, types of PSMs, has their own error rate. And so these need to be evaluated individually. So it's an additional step involved in the validation process. In this case, for every data set that you analyze, you don't run a single validation process, you run 4 validation processes. That's all you have to do is separate these types of PSMs and analyze each of them. Kojak does the separation for you. The validation is done using an algorithm that's not included in the TPP, but we've included it in this course as part of the tutorial, so you'll get to use it. It's called Percolator. It's a support vector machine that its design is actually amenable right out of the box to taking cross-linked PSM results, which is why we use it. It's applied to each PSM type. There are about a dozen different parameters that it accepts, and so it has this n-dimensional hyperspace that tries to find the optimal separation between targets and decoys, creates a hyperplane through that that is used to define your FDR cutoff, and then you set your threshold, say 1% or 5% FDR, and say, I will accept all peptide spectrum matches that are below that threshold. There are still, despite all of these efforts in validation, there are still some pitfalls that occur. One of the most common pitfalls is the occurrence of large peptides linked to small peptides. As explained with the algorithm, the peptide score, the score for each individual peptide is dependent on the number of B and Y ion matches that occur. A small peptide has very few B and Y ion matches, so there is actually very little spectral evidence for these peptides when they are linked to a large peptide. Therefore, if we look at the actual score contribution of each peptide in the analysis, the large peptide is going to contain the bulk of the score. The small peptide, very little. This very small score contribution might mean that it doesn't make it into our 500 or 1,000 peptide list cutoff in the two-stage analysis, or it could mean that some other peptide that is also small and similar mass might have a very similar score and therefore not create much separation in total score between this correct PSM and some alternative. And so all of those make analysis difficult, and these are common problems for everyone. The percolator algorithm does its best to solve these. We actually, as one of the parameters, the score contribution of each peptide is included as a parameter to help identify when these PSMs are correct or false. Other pitfalls. Intraprotein crosslinks are common in a sample. If you have a purified sample of a complex containing 3 proteins, chances are the bulk of your crosslinks are going to be to the same protein within that complex. That the actual cross-linking between 2 of the proteins is rare relative to linking within the same protein. However, mathematically, intraprotein crosslinks are very rare. So this means that your actual sample might have lots of intraprotein crosslinks, but the number of decoy combinations that are mathematically possible to replicate those intraprotein crosslinks is much smaller, so there is a bias towards your target database. That is the reason why intraprotein crosslinks are analyzed independently of interprotein crosslinks and loop links, and that helps solve this bias by creating its own analysis. Loop links have their own problem in that they are generally very rare. Usually when you have a crosslink, there's going to be a lysine or arginine between the 2 link points so that you have a true crosslink instead of these loop links. So because they are so rare, usually there is a very low number of PSMs in the results. The low number of PSMs means that there might not be enough information to produce accurate distributions, and so Percolator will actually give you a warning message when you have insufficient PSMs and when its FDR analysis might be inaccurate. The Kojak software is freely available independent of the TPP, although Luis explained yesterday how to include it into the TPP for the purpose of the tutorial. But if you ever want to explore Kojak more on your own, this is the website. There is, I don't have another slide, but there's a menu across the top here where you can get documentation, parameter information. Updates are generally monthly with new information, new releases, and the documentation is always evolving as questions come up and new analyses are discovered. And so the community is encouraged to participate in the development of Kojak. All right, thank you.